Hi, I'm a member of the Norwich Research Park and University of East Anglia iGEM team in 2012. We thought we'd do a quick video to try and explain our project because we've had a lot of support with Twitter and Facebook and stuff like that, but obviously people don't know the project just yet. So we thought, here we go, we'll try and explain it. So please bear with, and if you've got any questions, tap away at the comments section. So essentially we're working with biological sensors. The way they normally work is we've got a chemical of choice called the substrate. So it could be like caffeine, it could be a sugar, it could be a nitrogen species, which is what our project's on. That attaches to a section of DNA called a promoter, which then activates a section of DNA called the reporter, which produces a protein. So essentially, it's like a chain reaction. This activates this, that activates this. And the reporters use something like green fluorescent proteins. You've probably seen things that glow green when you add certain chemicals to them. That's going to activate something like this. So here's how it looks on DNA. Substrate comes in, DNA gets read all the way along here, and produces this. The main problem we have in biology is the fact that these promoters aren't particularly specific. So anything that's quite similar to this substrate can activate it, which makes it quite a problem if you're trying to work out specific things. So for instance, all of these will cause the cell to glow green, or red, or anything else. We're working with nitrogen species. So as you can see, this is an example promoter for nitrogen species, and there are 6 different things that could activate this one in particular. So it's not very specific, whereas we just want to work with this little one here, called nitric oxide. So what we're doing is we are looking to use lots of different sensors, which can then interact and give us just the final one. So for instance, you've got 3 promoters here. You've got the triangle, the circle, the square. The triangle is one of the rare promoters that has just one substrate. The circle has 2 and the square has 3. So for instance, you could take a readout, or you could use the amount of this against the amount of this to cancel out the reds and just have the purple. And then you can use all the amounts together to cancel out the reds and reds and purples from this one and just show the blue. So here's how it looks in a DNA form, as it were. So here's our different promoters. Here's our different reporters. We've got a section of DNA in here, which is to do with the protein coding. So for instance, if you look down here, we have, the way it works is you've got the promoter gets activated, so in comes one of the substrates. That recruits something called the ribosome, which is like a little bit of protein. That reads this, it's sort of like a barcode scanner sort of thing, and this is like a barcode. And then at the end, it produces the protein that we want. What we're going to do is basically take these, or these rather, and have these all cancel each other out so that the only protein that's produced is activated by one substrate, essentially. So for instance, we've got an example here. So you've got the circle and the triangle. The idea being that this section of DNA, we've designed that completely new and synthetically to interact with one another when they're expressed in the cell, so that these cover each other up like this. The ribosome can't get in there, which means no proteins are produced. And they cancel one another out. So for instance, you could have 5 triangles produced, 10 circles produced, 10 circles produced, and then the 5 triangles will cut out 5 of the circles, so these will get cancelled out, and you're left with just 5 circles. And that, so for instance, the triangles that are red, they cancel out the rest of the reds and produce just the purples. And that's how you can tell what the purple is. So, and this idea is completely brand new. That's our main project, is the idea of it interacting with one another, so it all happens in the cell. Normally you'd have to use some sort of equipment and it takes a long time. This should be very instant. And our idea is that we could use it physiologically in the body. So for instance, our project is using nitric oxide, which is this one, and nitrites, which are this one. We've got one promoter that does nitric oxide and nitrites. We've got another that just does nitrites. So the idea being that all the nitrite promoters will express this. They will cancel out all the promoters here, the DNA that's been expressed by nitrites, and this is showing just the DNA that's been expressed by the nitric oxide. At the moment, we're just working with fluorescent proteins with our reporters. But in the future, you may be able to use, for instance, a drug here instead. So, for instance, nitric oxide is quite prominent in cancer cells versus other cells. Therefore, cells that have a higher amount of nitric oxide then this system will produce more of this DNA, and this could be, say, an anti-cancer drug, and that will kill the cell that it's in. And that's basically our project in a nutshell. If you've got any questions, hit the comments, tweet us at NRP iGEM, and we'll be happy to answer them.